Welcome back, students. This is the first video on chapter 14. Chapter 14 is basically following electrons as they empower the cell to capture energy and utilize that for other reactions. The learning objectives for chapter 14 are varied, but they all encompass the movement of material supplying electrons to the electron transport chain. So the common theme is the electron transport chain. Please be familiar with these learning objectives. Photosynthesis is a process by which energy from sunlight is captured by plants, certain algae, and certain bacteria to generate food. And a byproduct is oxygen. So here's the general equation for photosynthesis. Carbon dioxide plus water in the presence of energy is converted to glucose and a waste product called oxygen. Those of you who are attentive will notice that this is the reverse reaction for cellular respiration. And indeed it is, although the enzymes and the place where this takes place are slightly different. Photosynthesis in plants occurs in chloroplasts, where cellular respiration begins in the cytosol and concludes in the mitochondria. Students should be familiar with how plants operate when it comes to photosynthesis. In most cases, water is available through the roots of plants, and carbon dioxide enters from the atmosphere in pores in the leaf known as stomata. The movement of sugars produced by photosynthesis is throughout the entire organism. So the leaves produce sugar, which is shared within the leaf and then passed to other structures, including the roots. And the roots absorb water and other ions and metals which are necessary for the workings of the enzymes in photosynthesis. Water is involved in supplying electrons to the photosynthetic mechanism, as we'll see, and extra water is lost through the plant's stomata during daylight hours due to heat evaporation, a process known as transpiration. For students not familiar with chloroplasts, chloroplasts are organelles. Organelles are found inside plant cells, and those plant cells make up all the structures within the leaves of a plant. The entry of gases, carbon dioxide and oxygen, is controlled by guard cells which make the stomata opening. Air spaces between the cells of the leaf allow the movement of those gases to the surface of those cells, where the gas dissolves into the aqueous environment and then is available as a dissolved substance for the chloroplasts. Within the chloroplasts, what we want you to understand is that two independent processes take place, stage 1 and stage 2. And both those stages are part of photosynthesis. So in stage 1, light energy is converted to chemical energy. That's the main purpose. It is quite sophisticated, as we'll learn, but that's what happens. And in stage 2, that energy that was generated through stage 1 is used to combine atoms of carbon dioxide into sophisticated molecules like sugar. This figure, taken from a general biology textbook, illustrates that to the next degree. So we have the light-dependent reactions, stage 1 of photosynthesis, which generates ATP, the high-energy molecule, and an electron carrier called NADPH, which carries electrons and protons. These two entities deliver their cargo, which is energy, to the light-dependent reactions, better known these days as light-independent reactions, but they have many names in many textbooks. In this case, they call that the Kelvin cycle directly. Usual choice of words as appropriate, any one of those three will work. And once the cargo is offloaded to drive this cycle, those energy carriers are then recycled back to the light reactions, the light-dependent reactions. So that would be ADP and NADP+, the exhausted version of those molecules. The entry of water is necessary for the light-dependent reactions, whereas carbon dioxide is delayed, and that's needed for the Kelvin cycle or the light-independent reactions. The two products of photosynthesis are oxygen, which is normally a waste product for the chloroplasts, and a small 3-carbon sugar, which is then able to be converted to anything that the cell needs to form structures for itself, for other cells within that plant organism. The term carbon fixation refers to the process where carbon in carbon dioxide is combined with other carbon to form more sophisticated molecules that are no longer gaseous carbon dioxide. So let's take a second to put in perspective what happens with terms of ATP and the electron carriers. So looking at ATP first, ATP is generated within the chloroplast through a process known not as substrate level phosphorylation, but an alternative way known as photophosphorylation. And we're going to explain to you what that means very shortly. During photophosphorylation, the energy from light is used to drive the transport of electrons through electron transport chains, which result in protons being pumped and then ATP being synthesized. That's the main learning objective of this entire chapter, that students understand how this process of photophosphorylation takes place. With respect to the electron carriers, NADP becomes charged with 2 electrons and a proton, and that's called a hydride ion. And where in the molecule is that energy captured? Well, this is the structure of NADP. And you can see in NADP, the hydrogen ions are carried right here in the uppermost ring, away from the phosphates. So phosphates are not lost, but electrons and protons are carried as passengers. Students need to become familiar with 2 mechanisms that have evolved on Earth. One of them is called oxidative phosphorylation, and the other mechanism called photosynthesis. Now, the 2 mechanisms do share a lot of their biology and chemistry, but there are some major differences. This figure from your textbook does a good job of illustrating that. So looking at the left-hand side of this figure, for animals and creatures that obtain food pre-built as molecules, fungi and some protists, the molecules are held together by covalent bonds, and those covalent bonds carry a lot of energy. So when the cell dismantles those covalent bonds, as we saw in glycolysis, energy is released. That released energy can then be used for building a gradient of protons on 2 sides of a membrane. So energy from food is transferred into a gradient difference on 2 sides of a membrane. And because this gradient has an unequal distribution, that is storing energy. So the energy of covalent bonds is actually stored now in the form of this gradient. Using oxygen to break down the food molecule completely, we have the process of oxidative phosphorylation. On the other side, we have photosynthesis. Photosynthesis relies on light energy. That's the photo component. So without light energy, this process cannot continue. So during daylight hours, plants and some bacteria and many algae are able to capture light from sun and convert that energy into the same gradient of protons on either side of a specified membrane. And within photosynthesis, we have a complex sequence of events that we need to learn. But the process is very akin to what's happening over here. Electrons are used to shuttle energy in both systems to power the movement of protons. And once those protons have been established, then later on, the gradient of protons can be used to generate work, and that work can be converted to the recharging of ADP into ATP. So the rechargeable battery of both plant cells and other creature cells can be re-established using this gradient of protons. So it's a two-step process. So first you break down the energy in food, or capture the energy in sun, give that energy to electrons, and then those electrons then power the movement of the protons. And then in the second phase, those protons are then allowed to re-enter the lower compartment, generating ATP. This figure does a great job in itself of illustrating that two-step process. So step one, known as stage one in this case, is the generation of energy from food or from light energy to move electrons, and those electrons then power proton pumps, which pump protons to one side of a membrane, and that stores energy in that gradient. That's called the membrane potential that we learned about before. And then the membrane potential allows the work to be transferred to the generation of ATP through enzymes that can synthesize ATP from ADP. And the power comes from allowing the protons to move back across that membrane. The textbook uses the word chemiosmotic coupling, and this chemiosmotic coupling is an ancient process developed by life a long time ago and still preserved to this present day. And the most notable thing to understand is that chloroplasts and mitochondria are the two structures able to perform this inside eukaryotic cells. Outside eukaryotic cells, many bacteria do this using their plasma membrane, as do the archaea. The textbook goes to great pains to get students to understand that mitochondria and chloroplasts are more like bacteria than they are like eukaryotic cells. One of their features is that the way that they divide. They are able to divide independently of cells, sensing when a cell needs more mitochondria. The mitochondria will enlarge and then split in the middle through a process called fission. And fission is something that bacteria do currently when they divide. And the other things that make us understand that these are ancient microbes that have now been sequestered is that they contain their own DNA. And that DNA is circular, just like modern day bacteria. And it contains genes that encode biochemistry, and that biochemistry is very similar to bacterial biochemistry rather than eukaryotic biochemistry. And the number of copies of the DNA inside both mitochondria and chloroplasts is greater than a few. So normally a number of these molecules, up to 20, are found inside these structures. The ribosomes that are produced are encoded mostly by the genes on the DNA of the organelle, although imported proteins from the cytosol are necessary to complement the missing proteins that are no longer part of the DNA of the organelles. Let's focus our attention on the anatomy of a mitochondrion. We'll focus later on chloroplasts. So the first thing to understand is that there are two compartments in a mitochondrion. The matrix is the gel-like material deep inside the heart of the mitochondrion. But then there's this white space which is between the two membranes. So mitochondria have an outer membrane that faces the cytosol, and then they have an inner membrane which faces the matrix. And the inner membrane of mitochondria is highly folded into these cristae, and that increases the surface area upon which biochemical processes can take place. The intermembrane space is important, as we'll see later, for storing protons. The outer membrane is permeable to many things below a certain size. The inner membrane, just like the plasma membrane, is more discriminating. In order to pass through that membrane, you have to have the right transporter, otherwise transport is inhibited. Not forgetting the overall scheme of things, this diagram here reminds us of the general architecture by which food molecules are handled by typical non-photosynthetic cells. So here we have sugar entering the cell, and then we have fats entering the cell. And the sugars enter the glycolysis cycle and are converted to pyruvate, whereas fats are dismantled into fatty acids and glycerol. And the fatty acids are then able to be imported across the inner membrane of the mitochondria using special transporters. And those fatty acids are then fed into the citric acid cycle as part of acetyl-CoA and other molecules. So here's the crux of the lecture. One way to remember what's happening is to immerse yourself in the biology. So I like to tell students that come to office hours, please make yourself into one of these electrons, and then you are then part of the journey and see where you end up. So initially you start life as a food molecule, glucose. So you are an electron within the glucose molecule. Eventually the glucose molecule is dismantled through glycolysis and the citric acid cycle, and electrons are pulled off. So you are pulled off your original food molecule, you're still vibrating with a lot of energy, and then you are handed off to high energy electron carriers. And NADH is one of those charged molecules. So NAD is the uncharged version that doesn't have U associated with it, and NADH is where you and your friends are taking a ride on that taxi cab, that high energy electron carrier. So when the charged molecule of NAD is generated during glycolysis and the citric acid cycle, you are a passenger on that molecule. Now, that taxi cab itself is no use to the cell unless it can unload its passengers to a destination. So you take a journey to a particular destination. Where's your destination? Your destination happens to be the inner membrane of mitochondria, where proteins receive you and then use your energy for migrating protons across membranes, as you'll see in the next few slides. So your energy is eventually utilized and you are then diminished in energy. So let the story begin. So here we have the migrating charged electron carrier, and it brings its electrons to its destination where it offloads its cargo, including you. And that cargo is then used for work. The uncharged electron carrier is then able to be recycled back again. So what happens to you? Well, here you are again being offloaded off of NADH, and then you enter this sequence of 3 protein complexes in the mitochondria. And this happens on the inner membrane of the mitochondria and not on the outer membrane. So these protein complexes are positioned one after the other so that the first one picks you up, uses some of your energy to pump protons, and a lower energy U is then passed on to the next protein complex, which then has the capacity to use some of your energy to pump more protons. And then now you have even less energy, and then you're passed on to a third protein complex, which can take some of your energy and pump even more protons across the membrane. Now that you're fully useless in terms of energy, you're exhausted, that's you now here. You and a friend are then handed off to oxygen and a proton to make water. So eventually your life ends as a water molecule, as an electron inside a water molecule. And then that water can join the rest of the cell. So this journey is long and hard, but your destination is to become water. And that happens inside human bodies as well as the bodies of any creature that performs oxidative phosphorylation. Now you know where the word oxidative now comes in, right? The use of water, the formation of water through the use of oxygen leads to the oxidative part of that statement, and phosphorylation will become later on when this energy here is used to make ATP. So keeping the bigger picture in mind, the outer white space here is the cytosol of a cell. The outer circle here, this gray circle, is the outer membrane of the mitochondrion. And then we have the inner membrane of the mitochondrion here, and the matrix of the mitochondria. So you can see these proteins that shuttle you around have an orientation, and they always move protons into this white space, the intermembrane space. Those protons will then be allowed to enter back into the matrix through a channel in this enzyme called ATP synthase, and that charges ATP using the energy of that gradient. So this is ATP being recycled, and that ATP will then be passed through the inner membrane of the mitochondria, and it will then leave and join the cytosol, where it powers many processes. Indeed, the inside of the mitochondrial matrix can also use some of this ATP for copying its DNA and for establishing protein synthesis. But most of that ATP is used to drive the cell itself. The carbon dioxide, which is generated during the citric acid cycle, is toxic, and that leaves into the cytosol, and then eventually it will leave the cell into the atmosphere. The arrival of pyruvate and fatty acids has already been discussed, but what we haven't discussed is that some of the NAD comes from glycolysis, and it enters again through the membrane and joins the other taxicabs, the other electron carriers, in delivering their cargoes here. So NAD delivers its cargo to this dark green protein, which is in this sequence here. Unfortunately, we do need to know what those three protein complexes are and how electrons are moved between them. And this figure here gives us that knowledge. So the dark green protein complex has a name. It's called NADH, dehydrogenase complex. Why? Because NADH is delivering its cargo to it, and the process of dehydrogenation takes place. So once you, the electron, delivers your cargo here, that energy is utilized to pump these protons in one direction, in this case going north. You're then passed to the second complex, known as cytochrome C reductase, through this shuttle molecule, ubiquinone. Ubiquinone shuttles back and forth between the first and the second protein complex, and it carries electrons one way in the direction indicated by these arrows. So you arrive at cytochrome C reductase complex, where some of your energy is harvested to move protons again from one area to another. Now that you have a bit less energy, you're then handed off to the final protein complex, and that's called cytochrome C oxidase. And that's done by another shuttle molecule called cytochrome C. So it's an easy way to remember these three parts here, because they all have cytochrome C in their name, and the actual molecule with the full name is called cytochrome C. So reductase on one side, or oxidase on the other. When you arrive at the third complex, you just have enough energy to pump more protons to the other side. And now that you're exhausted, as I said earlier, you're then combined with protons and other oxygen atoms to form water. If you look carefully, the green proteins indicated here are actually pumping, using energy, protons from one side to the other. So if you were to label that as proton pumps, you would be correct. So all three of these are proton pumps. Why are you moving protons across a membrane? Well, that's where physics comes in. The reason for moving protons is to store some type of force disparity across the membrane. So this gradient of protons is necessary for driving other biochemical processes, including the production of ATP, but also many more things. Because you're moving protons, and we know that protons are associated with pH, naturally there's a difference in the number of protons on either side of the membrane. Therefore, there must be a difference in the pH. And that's true. The matrix of the mitochondria, the inner liquid jelly-like substance, has a pH close to pH 8 because protons are depleted by being moved by those proton pumps to the intermembrane space. Because the intermembrane space contains a lot of protons, its pH is closer to 7, pH 7.2. And that's very similar to the pH that you find on the other side of the outer mitochondrial membrane, which doesn't prevent these protons from escaping into that environment. So we have this disparity in the proton gradient, and that establishes something called the electrochemical gradient for the cell. Now, as you know from another chapter, the electrochemical gradient consists of two parts. It consists of the chemistry as well as the electricity. So the chemical part is the driving force that's pulling the protons back again because they're higher in this space and lower in this space. And there's also a electrical force that's repelling the positive charge on these protons again down into the negatively charged space. So these two forces can be added up because they're facing in the same direction with respect to the protons. And therefore we have this huge electrochemical proton gradient. That's an additive effect of this and this. So that's called your proton motive force. The proton motive force. One of the most important protein complexes embedded in the inner mitochondrial membrane is an enzyme complex. It's called ATP synthase. And here's a depiction of what it looks like. It actually does consist of a rotary arm that spins, as well as some stationary elements that are held in place in the membrane and prevented from rotating. As the protons travel down their proton gradient, they cause this rotor ring to spin, and as it spins, it moves the stationary parts of the ATPase head back and forth because of its bulge here. And that molecular motion, that back and forth motion of those polypeptide chains, is enough to mechanically charge ADP into ATP. So there's a mechanical effect going on here. Even though this is a chemical gradient, the chemical gradient is translated into a the chemical gradient is translated into a mechanical effect. So this is a mechanically rejoining phosphate to ADP in a sophisticated manner. This is a real type of cartoon that illustrates how the proteins are arranged. So the head and the stalk and the rotor are interconnected in a very sophisticated manner. But the protons do travel, as indicated by the pink line, in that direction. One thing to understand, that just like any motor, if you reverse the motor, then you can generate the opposite. So in some situations when you have a large gradient, you can generate ATP. But you can do the reverse too. You can take the ATP and run the motor in reverse, therefore moving the protons back up again. You might think, well, that's a waste of time, but some cells, when they have extra ATP, they can drive this gradient for another day. So returning back to our overall schema, where we have the mitochondria inside the cells, each mitochondrion has the ability now, as you can see, to move food molecules, to move protons, to move inorganic phosphates, and to move ATP and ADP in different directions using transporters embedded in the inner membrane of the mitochondrial. This table provides us with an understanding of how many ATP molecules are recharged based on the transfer of energy. Let's look at the processes. So glycolysis, the breakdown of glucose to pyruvate, produces 2 NADHs and 2 ATPs. Now those result in the formation of 3 ATP molecules and 2 ATP molecules. Now these are made not by this ATP synthase, these are made directly by substrate level phosphorylation. So these ATPs are never made inside the mitochondrium, they're made in the cytosol directly from the energy of food. So there's a difference right there. But moving down, we know that pyruvate is oxidized to acetyl-CoA, and that 2 NADH molecules are charged in the matrix of the mitochondria. And those two, they generate 2.5 ATP molecules each. 2.5. So 2 would generate 5. The citric acid cycle generates 6 NADHs and 2 FADH2s and 2 GTPs, which eventually form 2 ATPs. So here we have a direct association. So these GTPs are again substrate level phosphorylation. So they make ATP directly. So there's a direct relationship with a small loss of energy, but they still make 2 ATP molecules. But these electron carriers here, this electron carrier, this electron carrier, this electron carrier, and that electron carrier, they cannot make ATP directly, but they can deliver their electrons to the protein complexes on the inner membrane of the mitochondria, the ones we just spoke about. And they, in turn, produce a gradient of protons, and that proton gradient will generate the appropriate levels of ATP. So those 6 NADH molecules that are made in the mitochondrial matrix through the citric acid cycle, they will generate 15 ATP molecules. And the 2 FAD molecules, they only generate 3 in total. So why do we have less here than we have here? And the answer is simple. Depending on which of the protein complexes receives the cargo from these taxicabs, then the appropriate amount of work is done. So NADH always delivers electrons to the first of those protein complexes. Which are we talking about? So let's go back. So here. So NADH always delivers its cargo right here. And therefore, all 3 complexes are used during the movement of that electron. And that produces a maximum proton gradient. Unfortunately, FADH2 delivers its cargo right here. And therefore, only this pump and this pump are available to those electrons. So that electron carrier has no permission to dock here. It can dock and leave its electrons with ubiquinone. So students need to remember that difference in the chemistry. Let's take a second to look at the ubiquinone molecule. Only the top part of the molecule is of interest to us. The rest of the molecule is down here, and that's your hydrophobic hydrocarbon tail. And you can look at that in another textbook. So this ring structure here is where the electrons are either relayed from one protein complex to the other, or this is where FAD unloads its cargo. And you can see electrons and protons, 2 of each, are handled by this ring structure. So when you combine an electron with a proton, you end up with a hydrogen. When you combine a second electron with a proton, you end up with a second hydrogen. So the ring structure here can accommodate 2 hydrogens as cargoes as it shuttles back and forth amongst the various proteins. The next figure is my favorite because it illustrates how energy is extracted from the electrons. So you, as an electron, are carried at a very high energy inside NADH. As you're unloaded, you enter a dehydrogenase complex, NADH dehydrogenase, and some of your energy is used to power the movement of protons. So naturally your energy diminishes. Then as you're shuttled to the next complex, some of your energy is removed for the maneuver of moving back and forth, and extra energy is taken from you to move protons. Now your energy has become pretty diminished, but still, you still have quite a lot of energy, and that energy is used to move you across cytochrome C into cytochrome C oxidase, where a large amount of your energy is finally extracted to move more protons. And now you're down here with very little energy, but still enough to just make you interact with oxygen and some protons to form water. And water has some of the lowest energy of all molecules used by biological systems. So high energy electrons, low energy electrons. So you have taken a journey from the top of this chart to the bottom, and your free energy has been extracted, right? So you have 0 energy by the time you get down there to do any kind of work. So that's why I like this diagram. The textbook wants students to understand that carrying electrons is a pretty sophisticated maneuver. And proteins that are able to carry electrons, they tend not to do them directly on the amino acids, but with prosthetic groups or other types of cofactors. So here we have an iron atom that is at the heart of this ring, which is part of cytochrome C. And you, when you take your journey, are captured by this ring here, which makes it very efficient to pick up and then deliver cargo in terms of electrons. So once you're shuttled across by cytochrome C, you end up with this complex called cytochrome C oxidase. And this too has in its heart a heme group, and the heme group is able to interact with electrons using the copper component. So now you can see where iron and copper are needed in the diet to keep these protein complexes operating at the right conditions. So in conclusion, what has this part of the chapter all been about? It's been about the electron, how the journey of the electron is useful for transporting energy from one location, food molecules, to a different location, a gradient of protons, and how that electron finally ends its journey as part of a water molecule. So if you keep that in mind, the second half of this chapter, in the next video, will make even more sense. But thank you for now.